from the Howard Hughes Medical Institute. The 2010 Holiday Lectures on Science. This year's lectures: Viral Outbreak, the Science of Emerging Disease, will be given by Dr. Joseph DeRisi, Howard Hughes Medical Institute investigator at the University of California, San Francisco, and by Dr. Eva Harris, Professor of Infectious Diseases at the University of California, Berkeley. The fourth lecture is titled Solving SARS and Other Viral Mysteries. And now, to introduce our program, the President of the Howard Hughes Medical Institute, Dr. Robert Tijan.
Welcome back to this final presentation of this year's Holiday Lectures in Science. It's a great pleasure once again to introduce Joe DeRisi to give our fourth and last lecture in this series. Previously, Joe told us about how using bioengineering, computers, and molecular biology, he's been able to combine these tools for a potent approach to hunt for new viruses. In this lecture, Joe is going to show you how he can use his viral chip in real time and in real life situations to discover and quickly diagnose new viral outbreaks. Joe will also, I think, give us a glimpse of what future in biotechnology holds towards the ends of his talk. And now a brief video about Joe.
Science as we know it now is a highly interdisciplinary endeavor. There used to be this concept that what was required was multidisciplinary involvement. You had different people who were experts in different things. You had the electrician, the machinist, the biologist, and the clinical MD, and the software programmer. And they spoke their own languages, and somehow they were able to get together and work on projects in a way that produced good science. Now, what's quickly been realized is that we can make faster progress, better progress, more accurate progress by learning each other's language, becoming simultaneously conversant in the multiple languages of engineering, computer science, and biology, and that individuals can do all aspects of the individual projects and not simply rely on experts who speak a totally different language and come at the problem from a completely different framework.
All right. So these are dengue native samples?
So there are advantages to being a specialist in one area or another, but there are also advantages to being a generalist. That is, being able to involve yourself in multiple different fields and innovate in different fields to bring it together. And this is especially true in biotechnology and biochemistry and virology and other kinds of sciences that require new technology development. Of course, what you hear in the headlines is the biological achievements that are made. A virus is discovered or so on. But behind all that is all the methodologies and technologies that were required to do that. And those don't necessarily gain the frontline headlines. But without the right tools, without the right technologies, looking into the unknown is all but impossible.
And we're back. Always wanted to say that. It's not as good as Mike Myers did it. So yesterday, I had the chance to tell you about different kinds of molecular biology techniques and tools that are used to develop diagnostics and tell if there's a virus in a particular sample and so on. And so today, what I'll be able to do is take you through a few examples of how we've used this in actual projects. And I hope to, by the end of the lecture, tell you about some exciting new technology that's coming down the road that will make sort of the things that we've been doing here today go even faster. So our first example takes us back to 2003. That's probably like when you were in, elementary school or something like that? It's kind of scary. But let's consider this real-world example. In early 2003, a mystery illness appeared, right out of nowhere, seemingly. And no one knew what it was. It was a respiratory disease, but no one really had a clue what was causing it, and it seemed pretty severe. And the sort of anxiety and panic about this outbreak was amplified further by the media, without a doubt. And some of the headlines that were coming out of these areas were pretty scary. It was called SARS, for severe acute respiratory syndrome. And by the time this reached its peak sort of anxiety and panic period, you couldn't go anywhere without hearing about SARS, and sales of face masks went through the roof. All right, so let's go to the heart of this epidemic or this outbreak. It actually started in mainland China, but our story really starts in Hong Kong, a special administrative region of China. And if we go Google style and zoom in on this outbreak, Hong Kong is here on the map. We'll zoom in a little further, and our story starts actually in one building, a particular hotel in Hong Kong. This is it right here. This is the Metropole Hotel. You can't get a reservation there anymore because it's not called that anymore. And you'll find out why in a minute. This story is centered on the 9th floor of the Metropole Hotel. And next I'm going to show you a floor diagram of the 9th floor. So this is the 9th floor of the hotel. The index case, the person who brought the infection into this hotel, that's the first person recognizable with the infection in an epidemic, stayed, ironically, in room 911. You can't make this stuff up. I mean, this is real. And now I'm going to show you on the floor plan where the other guests staying on that floor, who got sick. We already know the index case was sick. It was a physician traveling from mainland China. He checked into the hotel for a single night to go to a wedding and then left the next day. He was ill with this respiratory disease, but didn't really know the consequences of it. And people in these rooms and these rooms all got sick. And the ones in the orange, in particular, went on to transmit that disease to others after they left the hotel. So can somebody tell me, this pattern's not exactly random on the floor plan here. Can someone tell me why is it that there's this non-random distribution of hotel rooms around 911 that contracted the disease? Any ideas? The environment they were breathing in? So ventilation. Yeah. Ventilation, that's a great idea. That's one of the first things that you would look for in a hotel situation like this. It turns out that the rooms are generally under negative pressure such that the air is forced into the hallway and then into the rooms. So if the index case is in the room and breathing out a lot of, let's just say if this was transmitted by aerosol, which it really hasn't shown to be, then those particles would stay in his room. They wouldn't go out into the hallway. So that was probably not the cause. Maybe they had a big party. Is that one of your ideas? No. A cleaning staff. So perhaps the cleaning staff transmitted around. Interestingly enough, none of the cleaning staff got sick at this hotel. Any other ideas? One more.
The common use of the elevator and stairs.
Common use of some sort of pathway through the floor, for example, like the elevator or the stairs. I like that answer a lot. So it turns out that when this whole thing was said and done, this is caused by a virus. If it wasn't, I wouldn't be telling you about it. They went in and looked for the virus in the hotel to see what happened with this case. It turns out there was a massive hot zone of the virus. Tons of virus in the carpet directly in front of the room. So they could, 2 months after the outbreak, they could still find virus in the carpet. And so can you guess what happened here? Well, no one really knows. This patient died, we'll never know. But what likely happened is the person was sick and either coughed or vomited or threw up in front of the room on the carpet, embarrassed, doesn't want to tell anybody, cleans it up themselves, goes back in the room. And then people going to their room walk through it and get it on their shoes or whatever and go back to the rooms. Now, it doesn't explain all the cases in here. Maybe the guy in 938 got lost, went down the wrong way, went back down the other hallway, and that was a fatal mistake. So we'll never know. We'll never know. But this is the best that we can do in a retrospective study of a case like this of figuring out what happened. So SARS had this, it was just basically a case definition. It was anybody with a history of a high fever after a certain date, November 1st, 2002, because before that, SARS was totally unknown. a dry cough, breathing difficulty, and importantly, close contact with somebody else who had SARS. That was the case definition. What was scary about this? No diagnostic. No one knew what it was. Every diagnostic that was put on this thing was negative. The transmission mode was unknown. Was it aerosol? Was it something on surfaces? The incubation period was unknown. How long can people have it and spread the virus before they show symptoms? No idea. And the fatality rate at the time was actually pretty high. 14% is very high, actually. And that actually hides some details. It was age stratified. So patients that were 65 and older had case fatality rates of over 50%. And so that's an extremely deadly virus. All right, so now let's zoom out from the Metropole Hotel. There it is. We're back out into space now. And where did the people in that hotel room then go after they checked out of the hotel? They went all over the world. So they walked through the hot zone, they checked out, got on planes, and went home. And this was kind of a big deal. So the folks that went to Canada, one individual from the Metropole Hotel who went back to Canada then infected over 100 people at a hospital, directly linked to that case at the Metropole Hotel. And in fact, just to give you an idea of how efficient it was at spreading, in the hospital in Canada, the attack rate, that is the frequency that people who are exposed to disease actually get the disease, the attack rate for medical school students who entered in the room even to visit an adjacent bed of the index case was 100%. If you went in the room, you got SARS, which is pretty outrageous, unheard of. And so that's why SARS was scary. Now, this thing was just starting to go ballistic when one of the index cases who traveled to Vietnam checked into a hospital there, and the government in Vietnam noticed something funny about this case and contacted the WHO in basically late February of 2003. It said, we think we have a problem, which was exactly the right thing to do. They sent an infectious disease specialist, Dr. Carlos Urbani. He came to Vietnam, arrived in a couple days, checked it out and said, yes, this is something really different, and reported back to the WHO. So by March 8th or so, there was an emergency meeting at the WHO, and they said, okay, we got to do something about this. This is serious. Quarantine the place. Dr. Carlos Urbani called the shots, quarantined the place. All the health workers kept themselves inside and were not allowed to go home on their own choice. They didn't go home, which is a very brave thing to do. And that probably stopped the wildfire of SARS from spreading in Vietnam, unlike some of the other places that there were outbreaks. But he paid the ultimate price. Within a few weeks, he was dead of SARS. And he's a true hero of infectious disease and did the right thing. And it's a tragic story, but it's one of ultimate self-sacrifice. What ensued after that global emergency call was an amazing collaboration. Over 13 laboratories from around the world, 10 different countries at the minimum, came together with different researchers, technologies, and so on to collaborate on solving SARS because it was a global emergency at this point. And I'm going to tell you about our small part in this. We're not the only part in this. We were just one component, and I'll tell you what we did. So some of that sample from Hong Kong was sent to the CDC here in the United States. The CDC then distributed samples to various different locations, different labs in the United States, and we were one of them. So one of the samples came to UCSF. These samples were actually supernatants of a cell culture that had been inoculated with a piece of lung from a person who had died. We put that supernatant onto our virus chip. So I told you about the virus chip yesterday. You guys who did the bioinformatic exercise get to analyze some data and get some results. And if you remember from the bioinformatic exercises, we used a bioinformatic algorithm that we developed called ePredict, which basically compares observed signatures to known signatures to come up with the most likely result. And this is what Dave Wang, a postdoc in the lab at the time, saw when they did our little ePredict algorithm. He saw that the signature was best explained by a coronavirus from either birds, cows, people, and an astrovirus, a virus in a totally different family, mind you, from turkeys, from cows, more birds, and some human. Okay, that's a weird signature. It didn't explain. There's one, if it was just a human coronavirus, it should have just come up human coronavirus. But this was mixed. Now, a little bit of investigation reveals that certain astroviruses and a branch of the coronavirus family, the ones that are actually in birds, share a region that's almost identical. So astroviruses and coronaviruses have an evolutionary conserved region. And that's what our array was picking up. So this was a novel coronavirus. But I'm not a believer in this kind of stuff unless I see sequence. Sequence is the gold standard, like I told you. And so how are we going to get sequence? Well, we could do the, design primers, order them. They come in a few days, and then you do. This is an emergency. We want it now. So how are we going to get sequence fast? So a graduate student in my lab, Anatoly Urisman, had this crazy idea. He said, well, we know that SARS is on the array, right? So we hybridize this thing. It's sticking to the spot in the array. That's that signal that you see when you analyze those arrays. Couldn't we just take like a sharp needle and pick the spot off the array and sequence that material right there that was stuck on the end of the needle? That basically used the array to purify SARS nucleic acid. That's nuts, but what the heck? Let's try. So we got a neuron dissection rig from a neighboring lab in the middle of the night. We returned it. They never knew anything. A tungsten needle used to dissect neurons was used then under a microscope, confocal microscope, to actually scrape away one single spot that SARS had hybridized to. This is a rather insane little operation. But lo and behold, what we were able to do is just randomly amplify that material that's stuck to that spot. So we don't need any kind of specific primers, and just sequence a bunch of clones that came out of that. And sure enough, we confirmed a novel coronavirus. It was 65% identical to any known coronavirus, which means it's really pretty different. It was definitely new. And so we reported those results to the CDC and to the global response team. And around the same time, other methods like electron microscopy, other degenerate PCR techniques, and so on, all came together with the same conclusion, that there is a novel coronavirus here. And it is the most likely candidate for an etiological agent of the outbreak. And so what was amazing here is this all happened within about 1 month, the entire thing, from the time WHO found out to the time that this thing was essentially revealed to be a novel coronavirus. From the time we received the package to the time we got the result I showed you was 24 hours. So we were up all night, as you can imagine. And it was a really intense and exciting time. Now, what happened with the outbreak? So here is a histogram of the outbreak, the number of cases as a unit of time, just like some of the histograms that Eva showed. And here's where the novel coronavirus was discovered. So you can see from the beginning of the outbreak, the time it was discovered, to the time that effective quarantine procedures and diagnostics were deployed and control measures were put in place, and the epidemic was brought to heel and prevented from spreading further. So what could have been really horrible was actually put down pretty quick. Now you can see there's a little dip there. Does anybody have any idea what that dip is? No, it's not Carnival. No Carnival in sewer. I don't know what that dip is. Okay, I have a question to ask you. This was not a human virus initially. Most viruses that burn this quickly in a population they're not already there. We didn't know about this. Does anybody know what the reservoir of the virus is? That is its natural host, where it's not causing so much devastation. Any ideas?
Birds?
Birds. Well, that's a great suggestion because there's a lot of viruses harbored in birds, like influenza. But in this case, no. Any other ideas?
A cow?
A cow? Nope. But that was a good guess. I'll give you the answer. It was the horseshoe bat from central China. Now, I know it's a hard one, huh? It was initially thought that it might have been a civet cat, a kind of cat, but actually, upon further investigation, it turned out that the horseshoe bat had a coronavirus that's extremely close to the SARS virus, and only a few changes were required to then transmit it to humans. And I want to make a point here that many viruses that come freshly into the human population are so-called zoonotic transfers. They are the product of a transmission event between a non-human species to a human species through mutation or other changes. And so that is why it's also important to study viruses in different kinds of animals. And so the next story I'm going to tell you is an animal story, non-human, that's all about birds. And we heard, there was a question earlier that the bird might be the reservoir. Well, this is why we study some birds. So this story is about psittacines, though. Citizens are birds like cockatoos and cockatiels and macaws and parrots and things like this. Many of these birds are critically endangered. Some of them have less than 100 animals left in the entire species before they go extinct. And some of them are also very popular pets. They live for a long time. There's a very big domestic trade in these and so on. And for over 20 years, there was a disease spreading through citizens, over 50 species affected. This disease was called proventricular dilatation disease, or macaw wasting disease. It produced results like the one you see on the right. I'll get back to that bird in a little bit. It was 100% fatal. There were no diagnostics available. And for literally 2 decades, people beat their head on a wall trying to figure out what this thing is. There were several ideas, but none of them turned out to be true. And so how does this disease manifest itself? Well, here's a typical example. A breeder, an enthusiast hobbyist who breeds parrots and macaws in their backyard. Pretty common, actually. And what the breeder did, this is actually a true story. What the breeder did is he bought a new bird to breed with his other birds. So he bought an African gray parrot, brought it into the house, didn't know the bird had PDD, because a lot of times birds with PDD shed the virus before they're symptomatic. So that's why this one's colored red here. And put it in with his other birds. And what he attempts to do is breed it with the other birds. So he put in occasionally another bird. Well, the new bird, the African gray, stopped eating, which, by the way, is a sign and symptom of PDD. PDD is a gut motility disorder, which is why they waste away. And what was his response? The response was, well, maybe he's not eating because he doesn't like the other bird. Some sort of sociological pressure. So what did he do? He moved that bird to a series of different cages in the house, trying to find a better partner for that bird. Maybe it'll eat with this one, maybe it'll eat with that one. And you guess what's happening here. So he's progressively exposing other birds in the house to this one sick bird. Well, so the bird really wasn't eating, so the bird died. And soon after that, all the other birds that bird had contact with stopped eating. And you can guess where this is going to go. So pretty soon, he lost a pretty significant part of his flock to this disease. Now, we got on this case from notifications from veterinarians around the country and other places, and we established 2 specific collaborations, one with a veterinarian in Florida, Susan Clubb, and another in Israel, Adi Glantz. And we obtained samples independently from 2 different locations. That's also very helpful to do when you're investigating a mysterious disease, to make sure it's the same thing. These are little biopsies of bird crop, which is part of their digestive tract. We put those samples on the virus chip, not unlike SARS. And when I say we, this is really a postdoc in my lab. Amy Kistler is actually here today. Put it on the chip, let it hybridize, and she used ePredict and other tools to look at it. And one sample in particular really stood out. It stood out with this signature, just like you did yesterday if you were in the bioinformatic exercise. Borna disease virus was the first hit. The second hit was a human virus, respiratory syncytial virus, which is a very distant relative of Borna virus. They're both negative strand, non-segmented, single-strand RNA viruses. And so there's something interesting going on here. Because, why is it interesting? Borna virus isn't a bird virus. It's a horse virus. In fact, all known cases of Borna disease, which is caused by Borna virus, were in horses in a very narrow region in Europe. Outside of that, never seen it. It causes this disease called sad horse disease or Borna disease, and it makes the horses sad. But they're sad because they have all kinds of neurological symptoms and they don't eat very well. And sometimes they're called pipe smokers because they put hay in their mouth, but they never swallow it. And so this was interesting to us because possibly this could be a different Borna virus specific to birds, which would be a whole new branch of the family. And so Amy, pictured here, actually did a lot of molecular biology techniques like the kind I described yesterday. Make some primers, try and do some PCR, sequence some stuff. All came back pretty much garbage, with the exception of a couple little fragments that told us we're on the right track. We want the whole genome. I just don't want a little bit of it. And so after months of trying, could not pull this thing out because it was so different, which is probably why a lot of other people had trouble identifying it. So we tried a different technology. It's a new technology. I'll tell you more about it. It's called ultra-deep sequencing. So this is taking this whole sequencing thing to a different level. Ultra-deep sequencing is a way of doing a lot of sequence all at once. Instead of sequencing 1 template or 2 template, you literally can sequence millions of templates. I'll tell you more about what this means for biology in a little bit. But let me walk you through it. What happens is you take your nucleic acid, your DNA, your RNA, what have you, you chop it up into little pieces. They have to be little pieces because this deep sequencing technology is specific for sequencing short stretches. You then add primers, adapters. So these are little synthetic pieces of DNA that you either ligate or stick on the ends of the DNA. And that allows them to stick down onto a solid substrate, like a chip, like a DNA chip. Except in this case, you just flow them across this piece of glass, a flow cell, and they randomly stick down all over the place. And you end up with these little pieces of DNA stuck on the glass. Now remember Sanger sequencing? Well, this is a variation on Sanger sequencing. So now you have 1 million of these little molecules stuck all over the glass. Now, in one variation of the technique, they can be amplified in place to make a little colony. But let's just represent them as single molecules now for clarity. Bring in a sequencing primer, synthetic piece of DNA that's going to hybridize to the primer you stuck on the other DNA sequence. So you don't need to know what the sequence is you're sequencing. You don't need to make a specific primer here. And then you extend with the polymerase. But instead of using a bunch of nucleotides and some low frequency of a dideoxy terminator to terminate the strand, all the nucleotides are terminators. So it just goes one nucleotide and stops. And those nucleotides, those terminators, are labeled with fluorescent dyes so you can tell what they are. So then you take a picture of the flow cell, and you see a little colored dot, and that tells you what base got extended. Here's the magic. The next cycle, you can then cut off the fluorescent dye and reverse the terminator. So now it can be extended again. So reversible terminators. You extend with the second base, image it, look at the colors, cleave it off, and repeat. Third base, image it, and so on. So it goes extension, image, cleave, extension, image, cleave, over and over again. And you build up the sequence. You can do about 100 nucleotides this way. And so in cycle 1, 2, 3, and 4, if you look at the spot as it changes colors, you can read the sequence. So look at that one on the left. It's orange to begin with, then it's green, then it's blue. That's GTC, AAC, and so on. Now, how many spots do you think you can do? What does the real data look like? Does it look like these nice little discrete spots? Not so much. So this is actual raw data from the machine from my lab. Now, it looks hard to analyze. And that's why you have a lot of computers to do it. And there's a lot of informatics behind finding these spots. But the other point to make here is it's dense. This is less than 1% of the whole flow cell. And so instead of sequencing just a few templates at a time, we literally sequence somewhere up to around 300 million sequences simultaneously. That's kind of a big difference. And so and you might ask, well, if this is so great, why don't you just sequence everything? Forget the chips and PCR and all this stuff. Just sequence everything all the time with technology. Well, so it takes 5 or 6 days for this machine to run, and it takes around 3 months of computer time to analyze the data, and that's not really fast. And I'm not even going to tell you what it costs right now. Okay, so after you have all this data, what are you going to do with it? You got to assemble it. You got to put it together. Just because you have 100 nucleotide reads and a read being one little piece of sequence that are read from one spot, it doesn't mean you know what it is. You have to be able to put it together. So there's actually some sophisticated algorithms in bioinformatics that search for where the reads overlap and then combine them into what's called a single contig or contiguous sequence. And so one of the games here is being able to put all this stuff together. And I can tell you, we're not really good at it. Most of the algorithms out there, well, they're kind of bad. And they're getting better, and there's a lot of really smart people working on it. And so this is a tight marriage of bioinformatics, computer science, algorithm development, and technology. So what we did is we actually took all those reads from the Bornavirus sample and tried to map them to the known map of Bornavirus and guess where these things might map. And we ended up with a histogram like this. So a histogram showing you how many reads we have as per location in the genome. And what it shows is that we have sparse coverage. There's some areas which we have a lot of reads, then there's gaps and other things. So we can't really assemble the whole genome because we don't even know what's in there. And one of the main reasons is when we have a particular sequence, we don't have really good methods of assigning that to a virus. We may not know a sequence is from a virus because it's not long enough or has enough similarity to the known viruses, which has serious implications for using sequencing for discovery. Amy did is just take sequences right off the sequencer, though, and design those PCR primers overlap them, and recover the whole genome. So what was taking her months took her a matter of days after the sequence was in. Now, we've got the Bornavirus. Remember Koch's postulates? Does it really prove that the Bornavirus caused the disease just because I found it there? No way. There's lots of endogenous viruses and viruses that we get asymptomatically. And so while it's a good candidate, we don't know. Again, the microbe has to be present in every case. That's what association studies are for. You got to be able to propagate it in culture. That's not necessarily true in the modern age. But you want to know that the microbe causes the disease you say it causes. And so I'll cut to the chase here. With our collaborators, we were able to do an experimental challenge. We picked 3 experimental birds and 2 control birds. And for the experimental birds, they were injected with virus that was isolated from an infected bird. And then, in the end of the day, all 3 birds that were injected developed the disease. Now, we're blinded which birds were infected and which were not when we're analyzing the data, which is also important. The 2 control birds remained disease-free. Furthermore, from the infected birds, Amy was able to recover the Bornavirus in the newly infected birds, which is part of Koch's postulates. And for this reason, we believe that it really is the etiological agent, and this has been backed up by studies from other labs now. This is what the experimental bird looked like after it was infected. And what I'm showing you here on the right, you can obviously see the bird was wasting away and starving, but on the right shows the guts of the bird. And that stuff in there is undigested seed. This virus prevents food from moving through the gut. So the bird eats but can't move the food. And that's why they waste away. It is a neurodegenerative disease. It basically attacks nerve cells that pattern gut motility. And the immune system responds inappropriately and destroys those nerves. And food stops moving. Interestingly enough, there is a disease correlate in humans called idiopathic achalasia. Whether there's a Bornavirus involved in that or not, we don't know. But you can bet we're going to look. So, challenge study. Have we proved the cause of things? Association between symptoms of the virus? Yes. No virus in the healthy birds? Yes. We can isolate it from the sick bird, pass it to a new bird. We can re-isolate it. We can culture it. And we have association by other groups as well. So that's where that story's at. Amy's actually now culturing the virus as well. So I'll take questions there.
Besides dealing with unknown viruses, during your development of the ViroChip, did you encounter any other challenges?
In science, you always encounter challenges. That's a loaded question. One of the challenges that we faced initially was, how do we know this thing works? Because if you just take samples, sick guy off the street, you don't know if that's a virus or a bacteria or something. Where's your positive controls? And getting the right positive controls for the virus chip was always a problem. So actually, we were able to collaborate with a respiratory physician at UCSF, Dr. Homer Boucher, who actually had done a controlled infection experiment. We took healthy volunteers and infected them with rhinovirus as part of an asthma study under very controlled, safe conditions, approved by a human subjects review board and so on. And so this was the perfect positive control because we knew exactly how many viruses were going in. We knew what the viral load was in the patients as a function of time and so on. So teaming up with other clinical researchers allowed us to overcome some of these technical challenges. Oh yeah, sorry, I didn't see you back there.
Where do you think that the person who was in 911, where would he have gotten that disease if no one else had ever had it?
So he was not the first. As I mentioned, the disease actually started somewhere in mainland China. He was a physician and had been attending to patients with SARS in China. What hadn't happened initially was that it wasn't recognized early in mainland China as a new respiratory disease and reported before this big super spreading event that occurred at the Metropole Hotel. Question way in the back.
Has a treatment been developed for SARS?
Has a treatment, in fact, there's been a lot of people working on things like different kinds of inhibitors and so on as therapeutic agents for SARS. Some of that interest has died away because SARS itself has died away. Effective control measures and so on have sort of made the job of making specific therapeutics and vaccines to SARS sort of less important. But initially, during the peak of the outbreak, a lot of people got involved. And there were a lot of promising candidates. I think if it was still a problem, we'd see a robust drug or vaccine development effort if it was still an issue.
What makes SARS so much more transmittable than other viruses?
This is an interesting question. what makes the virus more transmittable? What makes it worse? Why is it so bad? Oftentimes, these zoonotic transfer events, that is when a virus comes from some other non-human species into the human species, it's not well adapted. That is a virus that goes too fast or kills too many of its hosts. That's actually not a very great virus, right? I mean, it's not great for us especially, but a virus that persists in the human population, that's allowed to replicate and spread through many, many individuals, are those viruses that actually do not kill their hosts right away, or take long, long times, have long incubation times, or maybe even go latent, like a herpes virus, which are some of the most successful viruses on the face of the planet. So SARS actually was kind of, in those kind of fitness contexts, kind of a lame virus, but scary nonetheless. And these are features of some of these viruses that come from non-human species. If they're given enough chances to infect lots of humans, they can adapt and become less virulent and perhaps persist longer in the population. Okay, questions are fun. All right, so my final story is one that's really a pleasure to tell you because it's a direct collaboration with Eva Harris. We're going to take you right back to Nicaragua. Still not going to dance. And the reason why we're interested in collaborating with Eva and others that have cohorts in different areas of the country is that it's well and fine to analyze respiratory secretions of individuals walking down the street in San Francisco, but let's face it, in different environments, you're going to see different viruses. People are exposed to different things, they have different hygienes, there's different community, water and sanitary structures, and of course there's socioeconomics, demographics, all that stuff affects everything. And so we teamed up with Eva because, as you heard before in her lecture, just well-established cohorts with kids that get yearly health visits, serum samples, and they're being seen for a variety of different things, not just dengue, like influenza and other respiratory disease. And so we're interested specifically in respiratory disease samples that were not influenza. It's got that covered. We don't need to do that. And so what Nathan Yaziak did, who's one of the TAs here for the bioinformatic exercise, is he went down to Nicaragua, helped develop some of those RT-PCR assays and other things. And what they did is started screening samples, respiratory samples, that is, patients that had a history of a cough or sore throat and had a fever in the last 4 days. Started screening those for different viruses. And out of these 718 samples that they did from one season, there's a bunch that were flu, there's some that were respiratory syncytial virus, there's some parainfluenza 1, 2, and 3. And so on. But a huge portion of those were unknown. Now, some of this is going to be bacteria. Some of it's not going to be a virus at all. So we know that. So what Nathan did is actually took 200 of those samples and threw at it not only the virus chip, but a whole bunch of other diagnostic assays as well. And out of this was able to assign a fraction of those to further viruses that we know about. Picornavirus, some rhinoviruses. But there was also a signature. When I say signature, a sequence, because remember, I only believe sequence in the end of the day. So he was able to sequence all the stuff that was positive by virus chip and PCR. And a small number of these had a new sequence for an enterovirus. Now, enteroviruses are interesting. I'll tell you why in a minute. He also tried to get the sequence, and like Amy, had a tough time because it was very divergent. So we brought out the big guns, did some ultra-deep sequencing. And these were 5 samples. Now, these 5 samples were from patients. When we look back at the clinical history, all of them had a history of fever and sore throat. That's how they got in the study. 2 of the patients were actually vomiting, and 1 had to be hospitalized. They were severely ill. So let me just take you through a little in the informatics. I'm not going to go through the algorithmic details, but just show you what kind of the data challenge it is. So only 1/8 of the output of the sequencer was used for this particular experiment, and that yielded 20 million sequences. So Nathan had 20 million sequences to read from, and this is from a respiratory secretion, a nasal swab from a patient. After filtering out stuff that he didn't believe was real sequence, simple repeats and other things that couldn't possibly be analyzed, he was left with 14 million sequences. After taking out the human sequence, because most of that, you swab somebody's nose, a bunch of it better be human, but I guarantee you it won't all be. He was left with 6 million sequences. So 6 million of that was not human. And then he said, okay, well, we know a bunch of bacteria. Let's take out all the bacteria we know. Now, there's a lot of ongoing sequencing of bacteria, and we were able to do the best we could is only remove about 2 million sequences. So we have 4 million sequences we don't think are human. We don't think are bacteria, at least to the bacteria we know of in the database. There's certainly plenty more. And now he went straight, together with Peter Skuse-Cox, another TA for the bioinformatic exercise. They analyzed those and asked the question, hey, which of these do you think are viruses out of that 4 million? And only 186 could we think were viruses. This is the needle in the haystack problem. And of those, only 119 did we think actually belonged to the picornavirus family, which is what enterovirus is. It's a picornavirus. Now, what are those other virus hits? It turns out that humans actually have a lot of endogenous retroviruses and things like that, and those are counted as virus hits, but they're part of your genome. That's another story for another day. So our perceived coverage, that where we thought these reads mapped to the genome, is shown here. This is a genome, sort of a typical genome map of a picornavirus, and so we have very sparse coverage. What Nathan did is he designed a bunch of PCR primers, a little more wild and crazy with the PCR design than the one I showed you for Amy, but nevertheless was able to recover the whole genome of the picornavirus. And after it was said and done and sequenced by Sanger, closing all these gaps, we actually had many more reads, well, about twice as many reads of this virus in the sequence than we knew about. But we couldn't tell that. There was no bioinformatic method that we had at that time, and we're trying to develop more sophisticated ones, that was able to ascribe those sequences to this new enterovirus. That's an interesting informatic challenge. Are you getting the theme here that you need informatics to do biology? Biology is becoming an information science. If you're going to go into biology and you want to do graduate school there, I encourage you to take a programming class, a statistics class, some computer science. For real. Don't give me that look. All right. This is where enterovirus 109, as we named it, maps onto the phylogeny. This is the genetic relatedness of these viruses. There's a bunch of different families here. The enterovirus Cs are shown on the right. And just for reference, this is where polio is. So it's in the same subfamily as polio is. Rhinovirus is also over there, the common cold. And many of these viruses have very different phenotypes. Some can give you meningitis. Some can give you polio and paralysis. Some give you the sniffles. It's an incredibly diverse family. What's interesting about that arm of the family, that EV109, is no one's figured out how to grow them. None of those guys on that branch have been grown to date. We don't know. Does it use a different receptor? What kind of cells does it infect? Can't figure it out quite yet, and we're going to work on that. This is a plot of nucleotide identity of our virus versus 3 other viruses in the picornavirus family. And what it shows is that this isn't 100% identical. It's like 65% identical, meaning it's very different, not unlike SARS. There's not a lot of information in this plot. If you look a little more closely, though, and ask, let's look at little subregions of the genome, we find something very interesting. So taking a small window of the genome, maybe 400 bases, and moving it across the genome and asking, what is this most similar to in the database? We find that actually the 5' end of the virus is more similar to a whole other branch of the family. And the rest of the virus is what we thought it was, an enterovirus C. That is, the 5' end of the virus, where all the regulatory stuff, the stuff that makes the virus go, translation, replication, and so on, belongs to a different part of the family. And this is how viruses diversify. One of the ways is that they can recombine. If you have 2 viruses that infect the cell simultaneously, they can exchange genetic information. Polymerases can switch templates and actually then create a new virus that will be endowed with different properties, perhaps totally different disease phenotypes and so on. And so this is an exciting and interesting area to know what kind of virus recombination is really happening in the wild. And now we have an example from it from real individuals and real people. And the follow-up here, this is where the work just begins. I haven't proven to you that this causes the disease in those kids. We don't know that yet. But now we have a lead. We can look, because Eva collected serum from yearly health samples, we could develop a serological assay and figure out who gets exposed to this and when in their lives. Is it a childhood disease, or do you accumulate it as you grow older? Who gets it and where? And what kind of disease phenotype is it linked to? Do they all have vomiting, or is it, healthy individuals have it too? That has to be all worked out. And of course, the molecular biology. All right, so now what does the future hold? Now, I told you about ultra-deep sequencing and how this is changing things. You probably don't understand, though, or realize what kind of change this is. Let's discuss change in a context. All right, for this example, I'm going to use astronomy, or the use of telescopes, as an example to show you how big this change is. So do people know what arcsecond is? When you use telescopes and stuff, an arcsecond is the minimum angle that's required to tell 2 objects apart. So it's a measure of how good your telescope is. If you can tell the objects apart, It's better than one which you can't tell them apart. The human eye is about 60 arc seconds. You can tell things apart when they're 60 arc seconds apart. Now, this all changed in the 1600s when Galileo made a telescope. His telescope reduced that to 3 arc seconds, a 20-fold improvement right away. And so this was kind of a big deal, and that's why he's really famous. Now, much, much later, nearly 400 years later, it only got 3-fold better. So a big telescope on the ground, Mount Palomar, about 1 arc second. You can tell I'm stealing this example from NASA. This is great. Now, what do you think the Hubble telescope can do? Because, I mean, that's not on the ground. It's up in space. It's pretty cool. It can do 0.05 arc seconds. So this is pretty amazing. So there's a couple points here. Number one, the Hubble telescope is the single greatest increase in optical resolution since Galileo built his telescope. Nothing really happened for 400 years. That's a pretty interesting fact in it itself. It turned out to be a 1,200-fold improvement over 400 years. The Hubble is really cool. But this talk isn't about Hubble. This talk's about sequencing. So here's the amount of sequence you can generate for $1. Now, you have to pay more than $1, obviously. But if you reduce it to the cost of $1, this is how many bases you can sequence as a function of time. You notice how the curve is going exponential? In just the last 10 years, there's been a 50,000-fold improvement in how much sequence you can do for $1, which is unbelievable because nothing in technology changes by 50,000-fold in 5 years. Not semiconductors, not computers, storage, data systems, none of that stuff. So we're really on a different trajectory here. And what does that mean? It means we can view everything as sequence because now we have the possibility of literally sequencing everything. And that's coming soon. In fact, it's already here in many ways. Have any of you heard of personal genomics or genetic testing? Have any of you done it? No hands raised. Well, I can understand that. But I did it. All right. And so I'll show you some personal genetic testing. Here's what happens. Right now, you can go on the web and you can get a bunch of different locations in your genome tested for various different disease associations. Now, many of these disease associations are important and interesting, but a lot of them don't have a big fold increase or a hazard ratio or sort of risk ratio associated with them. But I'll just show you a few. So it looks like has highlighted red here on the screen is that there's something going on with atrial fibrillation and not so much on the other things. The personal genetic testing also told me my eyes are blue. Good to know that. And that I don't flush when I drink alcohol. Like I didn't know that either. So, but no, in all seriousness, this is the sort of results that are reported today with the kind of genetic testing that we have, and it's only going to get better. This right now is only reporting that I have a 1.2-fold risk over the general population for getting atrial fibrillation for European men of northern descent. Here's one for obesity. They say that I have an increased ratio, it's only about 1.1-fold, of being obese by the time I'm 59. So right now, the point is, it says that our genetic testing, while the technology is developing very quickly, the data isn't quite there yet. We sort of just have a rough idea of what's going on, and it's only going to get better. We're in the infancy of personal genetic testing, and it's going to happen. The cat's out of the bag. Now, I also want to make a point that all this stuff is just testing the human genome, but we are more than just the human genome. There's all kinds of things around us. There's a huge number of bacterial cells in our body. There are more bacteria in you than there are human cells. And there's viruses. We're only now learning that there's a whole virome, associated set of viruses that inhabit you, not just the endogenous retroviruses, but many other viruses that don't cause disease, and we don't actually know what they do. And so we want to look at these as sequence 12. You can imagine the day when you get sequenced and every bacteria and every virus you have in you gets sequenced, perhaps even as a function of time. And that's also going to have impact. And so what we now do with genetic testing and test 30,000 genes for different associations may expand to testing the whole human microbiome, which includes bacteria and viruses, the virome. And that space may be much, much larger than your genome. And the context of that conversation between the bacteria and the viruses and our cells and our immune systems and our lifestyles and the foods we eat and so on, that's where there's going to be interesting new discoveries, new ideas, new things coming along. And I hope that you guys get interested in this and come along for the fun. All right. questions? Oh, come on, don't be shy.
Even with ultra deep sequencing, there's still a limited number of base pair sequences you can actually put into the machine. Are there any possible new technologies coming out that are commercially viable that can sequence longer base pair strands?
You bet. The sequencing technology I showed is just one of many deep sequencing technologies. This one's limited to about 100 base pairs. And what they do to increase sequence coverage is just sequence more 100 base pair sequences. So for example, my machine can do about 300 million. I'm upgrading it in a couple months. Thanks, Cheech. To something that's quadruple that. And so you can imagine something that quadruples every year. It's going to get deeper and deeper. There are other technologies that allow the monitoring of single polymerases to extend a template. So they can actually watch with a microscope a single polymerase putting nucleotides into a template in real time, not with dye terminators. And that technology has the capability of reading much longer reads, maybe perhaps 1,000 base pairs, 2,000 base pairs, 4,000 base pairs. We don't probably even know the limit yet. And that's actually, that technology is coming online right now. Disadvantage is that you sequence less templates. More longer, they actually seek longer ones, but few total of templates. But like I said, all these things double or triple every year in this kind of time of change right now. There's one back over here, and then I'll get to the center.
You were talking about all the bacteria and non-endogenous viruses that live in people. When in your lifetime do those come in? Are you born with them, or? No,
actually. So for example, if we just take the GI tract, this could be a topic of an entire other lecture, holiday lecture. When you're born, you're born sterile. That GI tract has no bacteria in it. And I actually have a colleague at Stanford who has actually collected diapers from babies as they were growing. That's a pleasant experiment. I have kids, so I know. And then analyzed how the gut becomes colonized and with which bacteria. And more importantly, how the population structure of those bacteria change when you take an antibiotic. And whether the population comes back or how does it change. It's now believed that this population structure that's in your gut, for example, has an impact on your disease state and on your life. It's been associated with things like obesity, for example. And so that's really interesting, and a lot of that is going to come out soon. Okay, I had one in the center, way in the back.
Can you use the microchip to sequence HIV and find out where it's, like, how it's occurring in people and if there's a way to stop it or, like, help it? Anyway.
So the DNA chip technology that I described is really for detection. There are DNA chip technologies that can resequence a virus or detect whether there are changes in a virus, so-called quasi-species analysis. There are chips that exist like that. And in fact, there are a lot of people sequencing, using deep sequencers mostly, to analyze how viruses are mutating, like HIV. And this is important because as we develop drugs and deploy them into the population, viruses adapt, mutate, and change, often in response to these drugs. And we want to have sort of predictive values to know where the changes are going to occur and so on. So this is actually an intense area of research right now.
I was wondering, do people know why, or like have a reason why animal viruses become zoonotic? Like what mutations occur and why do those mutations occur?
Well, in many animals, the cells that they have, the cells we have from mice to people and so on, they're all based on a common evolutionary lineage. They share many components. And so for many viruses, the mechanisms which they use to enter cells and so on are very similar, just maybe a few amino acid changes away. And so it's not inconceivable that it just takes a little bit of mutation and a chance interaction with an animal, whatever that may be, to result in a transfer. And the more contact with animals in those kind of contexts, like, for example, when you're exposed to the blood of an animal or something like this, the more chance that's likely to happen.
But are there like specific causes for those mutations, like, I don't know, UV rays or whatever?
Right, so let's talk about mutation in RNA genomes, for example. So RNA viruses are some of the most mutable genomes that we know. And they, on purpose, have polymerases that are sloppy, that insert errors on purpose. Their machinery is built to mutate themselves. Just to give you an idea of that scale, rhinovirus or something like that, 8,000 nucleotides approximately. And during an infection of an adult, every single position of the entire genome is mutated in every infection. There's 1 to 2 mutations made in the genome per round of replication, and there's 10 to the 10th or greater viruses there. That's way bigger than the genome. So the virus is actually making every mutation at every position every time, looking for new opportunity. And that's part of the evolutionary strategy of those viruses. It's actually a really exciting field. And it's a wrap.
Well, Joe, that was a fantastic set of lectures. What an inspiring and, I would say, optimistic, encouraging close to the series. We certainly have tools that will at least allow us to hunt down these new viruses. The stories that you and Eva have told us really help us to connect what goes on in the laboratory to what all of us care about in the real world. And I think that's been a special feature of these lectures this year. Certainly, many of us remember the SARS epidemic and the hysteria that was sweeping the world. I remember being in the airport feeling very naked without my mask. But it's fantastic to hear how quickly the timing of finding out what the source of the infection was. So let me extend my final thanks to both Joe and Eva for this incredible, lucid, certainly high-energy presentations. And now I'd like to take a little time to give you a little sort of advertisement. Because, of course, for next year, we have another set of exciting series of lectures. And the title is a very interesting title. It's called Bones, Stones, and Genes, The Origin of Modern Humans. And this is going to be such an amazing topic, we actually couldn't find just 2 good speakers. We had to find 3 great speakers for this topic. And they're going to be John Shea of Stony Brook University, Sarah Tishkoff of the University of Pennsylvania, and Tim White of the University of California in Berkeley. So from all of us at HHMI, I extend to you a healthy, virus-free, happy holiday season. Thank you.